This video is for the general public. I'll explain how the current vaccines work and introduce you to the virus and the variants that are causing concern. I'm Irene Kuhn. I'm a cancer cell biologist. I earned my doctorate in biochemistry and molecular biology at UC Davis. During my career as a research scientist, I've used many kinds of viruses to make vaccines and therapeutics. One of those viruses, an oncolytic adenovirus, is currently in clinical trials for the treatment of colon cancer. So viruses can be amazingly useful, but they can also cause havoc. Since the pandemic began, I've been reading scientific publications and listening to updates from virologists, immunologists, epidemiologists, and medical doctors. I've distilled what I learned into this presentation and invite you to share it with others. So let's get started with this talk about the COVID-19 vaccines, the virus, the variants, and immunity. The image is of the virus that is responsible for this pandemic, and you can see why it is called a coronavirus. The pandemic has been tough on all of us, very tough, especially on those who became ill or lost loved ones, and on the people who risked their lives to care for us. When businesses closed, many lost their livelihoods. When schools closed, family stress increased. And overall, our social inequities shamed us. Here's what I'll cover. I'll focus first on the mRNA vaccines and the 40 years of research behind them. I'll discuss the efforts to ensure racial and gender balance in the clinical trials. I'll explain how vaccines generally teach our immune systems to protect us, and then summarize the studies of how well the vaccines are actually doing. The next section is about the virus, what we've learned about how this respiratory virus spreads, how it infects and takes over our cells, how it makes new, potentially more dangerous variants. And I'll conclude with why reaching herd immunity through vaccination is so important. These are the kinds of questions I began with and that I'll answer here. I wondered why I should get vaccinated. Were the vaccines safe? What about how quickly these mRNA vaccines were made and approved? Could the vaccine make me sick? Could it change my DNA? Could it kill me? Let me get that one out of the way right away so you don't have to worry about it. No one has died from being vaccinated with these vaccines. No one. If I were vaccinated, what could I do safely? And why do I still need to wear a mask? So you can follow along. These are the basic terms of this pandemic. SARS-CoV-2 stands for Severe Acute Respiratory Syndrome-Related Virus. That's the SARS part. It's a coronavirus, CoV, and it's the second one of this type, number 2. COVID-19 stands for Coronavirus Infectious Disease 2019. mRNA is the acronym for messenger ribonucleic acid. Spike is the big protein on the outside of the virus that pulls the virus into cells. This protein is required for infection. Looking at the graph on the left, the red bars indicate the proportion of deaths due to COVID-19 in each age group, while the black lines indicate the proportion of the U.S. population in that same age group. Death from COVID-19 is more likely the older you are. In the graph on the right, the left column shows the proportion of deaths from COVID-19 for each racial group, and on the right, the proportion of the U.S. population for each racial group. Clearly, the non-white U.S. citizens are paying a heavier price in this pandemic than those of us who are white. I got vaccinated once I had become comfortable that the vaccines are safe, that they will protect me from getting seriously ill or even dying of COVID-19. I also got vaccinated to protect other people, because becoming immune by vaccination is the only way to stop the creation of new and potentially deadlier variants of this virus that would prolong this pandemic. So how do vaccines protect us? Vaccines teach our immune systems to recognize infectious agents, such as the SARS-CoV-2 virus illustrated in the upper right-hand corner of this slide. This recognition by the immune system is very specific knowledge. It's like having a wanted poster of the culprit in fine detail. That specific knowledge is stored in your immune system's memory cells. Later, if you get infected, those specific memory cells very quickly reproduce and travel through your body to find and destroy the infectious agent, thereby reducing the possibility that you could pass the infection to others around you or get sick yourself. Memory cells are very long-lived, so they can protect you years later if this specific infectious agent appears again. We don't yet know how long the SARS-CoV-2 memory cells will be functional. We can only learn that as time passes. But we are encouraged as we now know that memory cells from the 1918 flu lasted at least 90 years. There are three parts or elements of good vaccines. The first is the message they send to your body. The second is the language in which that message is written. And the third is the delivery vehicle. It took over 40 years of research to figure out how to make excellent mRNA vaccines. I don't expect you to read all this, but I list it here to show that the research, the published research, on which the mRNA vaccines are based, goes back more than 40 years. The idea of these vaccines was carefully developed and tested and retested over many decades by many different, often competing groups of research scientists. Most of this research was funded by your tax dollars, supporting research groups at universities and national laboratories, such as the Infectious Diseases Division of the National Institutes of Health, and that includes the Vaccine Research Center. Also contributing to the great speed with which these vaccines became available to us is the unprecedented level of collaboration of scientists and research groups from around the world and global collaboration between governments and philanthropists that came together to fund companies so that they could produce millions of doses of vaccine candidates even before these vaccines had been proven effective. The damage of the pandemic engendered unheard of cooperation. Let's look at a few of the scientists whose decades of research resulted in the mRNA vaccines. We'll start with the vaccine now called the Moderna mRNA vaccine. As indicated by the photo on the left, the Moderna mRNA vaccine is actually a product of our National Institutes of Health's Vaccine Research Unit. You'll probably recognize Dr. Fauci and the former president, and to his right is Dr. Corbett. Dr. Corbett heads the mRNA COVID-19 vaccine team at the NIH. Dr. Kariko, pictured here, is one of the Hall of Fame scientists of this pandemic because of her 30 years of tireless work against great odds, trying and finally succeeding to make RNA vaccines work. Why was this work so hard? Because mRNA is very fragile. mRNAs are important molecules in every cell in your body. mRNAs encode the information that tell your cells to make the proteins required for life. And to regulate all that information, these mRNAs have to be destroyed as soon as they have delivered their instructions. So your cells rapidly eliminate mRNAs as soon as they are read. Because mRNA is so fragile, it took Dr. Kariko decades to figure out exactly what kind of mRNA bases could survive long enough to be useful and how to purify that mRNA in the lab so that it would elicit the desired kind of immunity. And then how to encapsulate those mRNAs in microbubbles that could be effective vaccines. Here's Dr. Kizzmekia Corbett's team at the NIH Vaccine Research Center. Dr. Corbett is the lead scientist of the strategy she calls the, quote, prototype pathogen approach to pandemic preparedness, unquote. Using this strategy, she and her team have for many years been making a whole range of vaccines against those viruses that we know are most likely to cause the next pandemics, and then evaluating immune responses and protections against those same viruses in animal models. It's dangerous work handling all those viruses, and it's also complex and laborious process of test, adjust, repeat. Successful vaccines are then tested in humans once they are safe. Over the years, this group has developed mRNA vaccines that had entered early phase trials in humans. These include vaccines for rabies, Zika, flu, cytomegalovirus, and respiratory syncytial virus. I asterisk the RSV, or respiratory syncytial virus, because it was the intensive research by the next two scientists that was critical to the ultra-rapid design of the COVID-19 vaccines. The blue arrow on the left points to Dr. Barney Graham, chief of the Viral Pathogenesis Laboratory and co-director of the Vaccine Research Center at the National Institutes of Health. Dr. Graham is surrounded by his research team. Dr. Graham has focused on developing a vaccine for respiratory syncytial virus for over 30 years. Respiratory syncytial virus is a common infection, but can, unfortunately, cause severe disease and even death in young children. Dr. Jason McClellan, in the photo on the right, is particularly interested in the structure of proteins. He joined Dr. Graham's lab about 10 years ago to work on the structure of the RSV spike protein. Resolving the structure of a huge metastable or floppy glycoprotein like the RSV spike requires very high-powered cryo-electron microscopes and a lot of computer power. Together, these two scientists resolved and, in 2013, published the RSV spike protein structure and also, importantly, figured out how to stabilize that huge protein in what is called the pre-fusion conformation. That is, the shape this protein is in on the surface of the RSV virus when it infects human cells. This version of the protein is called spike 2P. When Chinese scientists figured out and published the whole genome of the SARS-CoV-2 virus on January 10, 2020, Dr. Graham saw immediately that the spike protein of this new virus, the SARS-CoV-2, was very similar to the spike protein of RSV. That was a huge breakthrough. Between them, when the pandemic struck, these scientists had all the experience needed to rapidly design an mRNA vaccine. Drs. Graham and McClellan knew exactly which version of the spike protein should be the message. Dr. Carrico had published the language and a variety of nanobubble delivery vehicles that could be used for mRNA vaccines. Dr. Corbett's years of testing showed how to put the SARS-CoV-2 vaccine together, message, language, and delivery vehicle, to make the most effective, immunogenic, highly protective vaccine. The clinical trials of the vaccine were designed to test the efficacy of this vaccine to protect against serious and lethal COVID-19. Over 30,000 people who were at risk, that is, likely to be exposed to the virus, volunteered for the trials. The trials had to be effective for all races and genders in our population. Equity and inclusion in the clinical trials is critical. Much work continues to be done to gain the trust of people of color, as these groups have been abused by the medical and scientific communities. Review and management teams for the clinical trials included BIPOC scientists and doctors ensuring that racial and gender balance for vaccine clinical trials reflected the racial and gender balance of the United States. The Biden-Harris transition team, working in parallel, included experts from underrepresented groups. All of this is reflected in the next slide. This is a screenshot from an NAACP virtual town hall program available on the NAACP website. Note that Dr. Patrice Harris was president of the American Medical Association in the year before and during the pandemic. Dr. Nunez Smith, on the top far right, served as co-chair of the Biden Coronavirus Advisory Board. Dr. Corbett from the NIH was part of this panel of experts. She's shown on the bottom row, second from the left. So how well did the vaccine do? This is a page from the New England Journal of Medicine publication of clinical trial phase 3 outcomes. I've enlarged the graph on the right. The cases of serious COVID-19 illness in the placebo group kept rising over the 10-month trial period, while the yellow line shows very few cases in the vaccinated group. We have to say that adverse events are expected from any vaccination program, and we know that there have been a few. But we also know that COVID vaccines cause fewer adverse events than the flu vaccines that we get yearly. The next slides will look deeper into the Moderna vaccine, what it is and how it elicits a protective immune response. First, vaccine cannot make virus in you. The COVID vaccines can only make spike. Here's a cartoon of the SARS-CoV-2 virus. You'll notice those red things that look like golf tees sticking out of the virus surface. Those are the spike proteins we saw a few slides ago. The gene for spike is shown as a red coil, and it is the mRNA encoding spike that is in the mRNA vaccines. Notice that the gene coding for spike is only 4,000 letters or bases long. The whole genome of this virus is over 30,000 bases long, and all those 30,000 bases are required to make a virus. Since only 4,000 bases are in the vaccine, there is no way that the virus can be remade in your body. The message for spike cannot recreate the whole virus in you. The vaccine, encoding the mRNA for spike, teaches your cells to make copies of the spike protein that look just like the ones on the virus. That process is illustrated on the right. The vaccine is shown as the red coil of spike mRNA inside of the oily nanobubble that delivers the mRNA to your cells. These oily bubbles fuse with the fatty surface of your cells, and the mRNA enters the cell. The mRNA is taken up by ribosomes that read the mRNA instructions to make spike proteins. The spike proteins end up on the surface of your cells, ready to train your immune system. Once the vaccine is in you, when immune system cells, such as the B cells or T cells, bump into your cells that have newly made spike proteins on them, the B and T cells recognize the spike as foreign, and they learn to defend you against the SARS-2 virus. The B cells learn to make thousands of different kinds of antibodies that can glom onto any COVID viruses that may get in you, and the T cells learn to destroy any infected cells. This process takes about 2 weeks, so please be sure to wait 2 weeks after your last vaccine shot before you consider yourself immune. This slide is a reminder that T cells are not only a very important part of our immune response, but they are also a very durable part. The new variant viruses may be able to evade some aspects of our B cell-made antibodies, but are very unlikely to escape the killer T cells. In summary, B cells neutralize virus particles while T cells kill infected cells. All the COVID-19 vaccines teach your immune system to make thousands of kinds of antibodies to attack the SARS-CoV-2 virus and create swarms of T cells that kill any cell that contains virus. Importantly, vaccines teach your body to create both B and T memory cells. The populations of these cells will quickly expand so they can attack any future SARS-CoV-2 viruses that infect you. Next, a look at how well these vaccines are working. Clinical trials measure vaccine efficacy, but the real test is how well do the vaccines work in a general population. This is effectiveness. Israel was the first country to have vaccinated a large percentage of its population. They recently reported results of their study in which they tracked the health of 1.2 million people, half of whom had been vaccinated and half of whom had not been vaccinated. The groups were balanced for age, gender, and pre-existing morbidities. Note that 30% of these groups were over 60 years old. The results show that the mRNA vaccine made by Pfizer was 94 to 96% effective at preventing serious disease across all age groups, even those over 70 years old. This shows the vaccine protection does not diminish with age, and it's such good news. It is particularly assuring that the vaccine performed so well in spite of the study being done during the largest outbreak of COVID-19, during which the more infectious United Kingdom variant had become the dominant strain in Israel. The next two studies of the possibility of reinfection after vaccination were done on staff at two medical centers. Both studies used the PCR test so that they could detect asymptomatic carriers as well as those with apparent illness. The LA group tested their vaccinated staff weekly. You can see the numbers of new cases dropping, see the second column from the left, as time post-vaccination increases. The last line is 15 days post-second dose, so full immunity would be expected. And you can see that 7 people out of 15,000 people, that's all, were either ill or found to be carrying the virus asymptomatically. The Dallas medical staff reported 4 people were either ill or shown to be carrying the virus out of over 8,000 staff tested. This percent of protection is in agreement with the LA results. Both studies show around 98% protection. These studies show a very high degree of protection from mRNA vaccines. The Pfizer-BioNTech mRNA vaccine encodes the same version of spike, the spike 2P, as the Moderna vaccine, but in a slightly different oily nanobubble. The Johnson&Johnson and AstraZeneca Oxford vaccines are both adenoviral vector DNA vaccines, and Sputnik V from Russia is very similar. So again, these vaccines encode the same message, the virus's spike protein, but using DNA as the language instead of mRNA. The DNA is delivered inside of attenuated adenoviral vectors that are taken up by your cells. The outer shell of the vaccine is stripped off inside the cell. The DNA enters the cell nucleus where it is transcribed into spike mRNA, which leaves the nucleus. The rest of the story is the same as for mRNA vaccines. The spike mRNA is translated into protein. The new spike proteins migrate to the surface of the vaccinated cell, where your T and B immune cells encounter the new spike protein and start learning to mount an immune response against this viral protein. This type of adenoviral-based vaccine has been used before to fight Ebola and Zika virus outbreaks. The safety and efficacy of these vaccines have been shown. These adeno-based vaccines have some advantages over mRNA vaccines. Only one shot is needed to become immune. This vaccine is stable in a refrigerator for 3 months. The one and done and refrigerator shelf life make these vaccines excellent for use in rural clinics and in emergency rooms where many people receive their health care. Note that these adenovirus cannot replicate and they cannot integrate into your DNA. To summarize the vaccines section of this video, all these vaccines are destroyed in your body very quickly. None of these vaccines can change your DNA. They all encode the same protein, spike 2p. They use either DNA or mRNA as their language, and they use different delivery vehicles. People have voiced concerns about side effects. We know that there will be side effects. That is expected for any vaccination program. But experience with previous vaccines shows that any side effects caused by a vaccination will have been seen within the first 2 months after completion of the clinical trials, so we can be prepared for them. It is this knowledge that supported the FDA's decision to release the COVID-19 vaccines 2 months after the completion of their clinical trials. The release of the vaccines was not rushed. That said, the CDC continues to collect data on everyone who is vaccinated. You can contribute to this effort by signing up on the CDC website for the vSafe program. The uplifting news is that the current vaccines protect you 2 weeks after completed vaccination very well against even mild COVID-19 and apparently completely protect you from dying of this disease. Next, let's look at the SARS-CoV-2 virus. Here's my favorite representation of the virus. The spike protein reminds me of fried chicken drumsticks. To start with, viruses are not alive. Viruses must infect us in order to make copies of themselves, in other words, to replicate. When the pandemic started, we were all very fearful because we did not know how this virus was spread. How could it infect us? Some viruses can remain infectious on surfaces, that is, other kinds of viruses. So we sanitized everything: doorknobs, packages, mail, food. We stopped shaking hands. Luckily, tests soon showed that this particular virus is one that does not survive very well on most surfaces. So we could relax a little bit about sterilizing surfaces. But please continue washing your hands, because if you touch your face, you may get something into your eye. Once scientists isolated the virus, it was shown to be a respiratory virus. Many common illnesses, such as colds and the flu, are caused by respiratory viruses, but this one is deadlier. Respiratory viruses are expelled from our noses and lungs in aerosol sprays, or droplets when we talk, sneeze, or cough, or sing. We soon learned we can get infected by someone who is not sick. Because this virus takes a few days after we are infected before we feel sick, and during those few days, we are already contagious. This is a great photo showing the aerosols and droplets that spew out when we sneeze or cough. But note that his Kleenex or cloth stops most of the spray. That's why masks are so important. Wear them whenever you are out. You never know when you or someone around you might be contagious. This drawing shows 2 people standing close enough that aerosols and droplets emitted while speaking are landing on the other person. Social distancing keeps the spray zone away from you. Also note that your chance of getting infected while talking increases with the length of time you talk. Try to be brief. That's what it looks like by electron microscopy if your lungs become infected. The hundreds of virus look like spiky balls. It's not too pretty. A single virus infecting a cell can replicate 1,000-fold, releasing hundreds of infectious virus into your lungs. When you talk or cough, you can too easily infect the people around you. For a closer view, these images are from an animation showing how the SARS-CoV-2 virus infects cells. There's the virus in the upper left panel. This time the spike proteins are shown in green. The surface of the cell is below the viruses, looking like a gray rug. The ACE2 receptor, the one that the spike protein is looking to latch onto, is shown in purple, sticking out from the surface of the cell. Moving to the upper right panel, a closer view, here the spike proteins have found and latched onto their ACE2 cell receptor landing pads. What happens next is truly amazing transformation of the spike protein into a nanomachine that deploys tendrils down into the cell surface, pulls the virus surface smack on top of the cell surface, causing these two surfaces to fuse. Once fused, the virus shoots its genome into the cell. The bottom right panel shows the cell after the virus has entered it and moved toward the center of the cell. But note that the virus's spike proteins are left on the surface of the cell. Remember that vaccination teaches your immune system T cells to recognize infected cells and kill them. So the viral proteins left on the surface of this infected cell mark it for destruction. To sum up this brief section on the virus's spike proteins are the nanomachines that grab onto the cell surface and then pull the virus into the cell. The virus then takes over the cell, turning it into a virus factory. The virus replicates, producing thousands of virus. Viral surface proteins remain on the outside of the cell so that once you become immune, your T cells recognize those viral proteins and kill infected cells. But it is that massive replication of viruses that worries us, because that is when variants are created. Some variants have made the nightly news because they are more transmissible and/or they cause more serious illness. These variants are very rare, but every one of those variants was created when someone was infected, including possibly someone who is asymptomatically infected. This is true because viruses mutate, which is to say that they generate variants only when they replicate, and viruses replicate only when they infect someone. So as long as people continue to get infected, new variants will emerge. We worry that the vaccines we have may not protect us if the variants become so different from the original virus that our immune defenses cannot protect us from the new variants. Vaccines prevent infection. If a virus can infect, it can't generate variants. So that's how you can help stop the creation of new variants, by getting vaccinated. This point is so important that I will repeat it. Viruses are not alive. They cannot reproduce on their own. Viruses cannot produce variants on their own. Viruses are created only when viruses replicate inside our cells. That is, when we are infected. So when we get infected, the virus creates variants. Vaccinations reduce infections, reduce replications. When the virus cannot replicate, it cannot produce variants. Vaccines stop variants. Until you can get vaccinated, please stay safe and wear a mask so you don't get infected. As I am preparing this video, some people are vaccinated and immune and others are not. So here's how the CDC asks that we manage this. When fully vaccinated people are with vaccinated people, they can take off their masks and can have meals together, even indoors. If any unvaccinated people are in the group, masks need to be on to protect those who are not yet vaccinated. If no one is vaccinated in your group, or you don't know if there are unvaccinated people in the area, please keep all safe practices, masking, keeping distance, and frequent hand washing. These guidelines change as the pandemic changes, so check with your favorite news sources to keep up to date. New variants raise fears of increased transmissibility of the virus. Many people advise to wear 2 masks to double our protection against aerosols. So here's someone wearing 2 masks. Obviously, 2 masks completely overlapping. I won't read these off, but these are the resources I've used in preparing this video. They are public sites where you can get updated information. I particularly love the podcasts of the 5 or so scientists who broadcast This Week in Virology, affectionately known as TWIV, on Microbe TV website. Another wonderful web-available set of videos are those from Grapevine Health. These are the brainchild of Dr. Lisa Fitzpatrick. Dr. Lisa talks with people in the streets in her Ask the Doctor series. That's it. Long, but you've reached the end of this video. I hope you're more comfortable with the science behind the COVID-19 vaccinations. These vaccines are safe and effective, reducing severe disease to zip. It's amazing. Current data are looking very good, indicating that vaccination nearly wipes out transmission of the disease. Vaccination also nearly wipes out the chances that you can get reinfected by this virus if you are vaccinated. Obviously, we do not yet know how long this protection will last. So do not throw away your masks. Remember the shortages from last March? Masks are precious, especially after you've found one that fits your face. And on a final note, variants. Please remember that the only way to stop variants from cropping up and prolonging this pandemic is to get vaccinated.